So my name's Professor Nellie Georgiou-Christianus, and I'm a cognitive neuroscientist within the School of Psychological Sciences at Monash University in Melbourne. I'm also the Director of Research Degrees within the school, which means I oversee all the PhD students and the doctoral students that are conducting research within the school. So I was an undergraduate student at Monash University doing a Bachelor of Science, and in third year I was inspired by one of the professors who was talking about neurodegenerative disorders, and I became immediately fascinated by how the brain changes as a consequence of disease. So I remember following him after his lectures and asking him about disorders such as Parkinson's disease and Huntington's disease, and whether he would take any honours students, because I knew at that time I really wanted to do research, and obviously to do research one needs to do honours and then a PhD and so forth. I was then accepted into an honours program, and I undertook an honours program on Parkinson's disease, and during that honours year I was interested in how patients with Parkinson's disease could improve their motor dysfunction by the provision or using external cues. So people with Parkinson's disease have a poverty in movement, but during that year we found that by providing external visual cues, it acted as a prompt for the patient, so the patient was able to improve their motor function using these external prompts. When I started the PhD, I was interested in a rare neurodegenerative disorder and how I could make a difference in something that really no one else was researching within Australia. And so I made contact with a neuropsychiatrist at St George's Hospital, and he had access to patients with Huntington's disease. In fact, in the early 1970s, he established the first Huntington's clinic ever in the world, and so we already had a reputation within Melbourne as being as offering clinical care in Huntington's disease, but there was not much research that was being done at the time. So I made contact with him, and we then started a project on Huntington's disease. At that time we were looking at trying to understand the behavioural deficits in Huntington's disease using experimental tasks. So we were able to tap into things like attention, cognition, motor dysfunction, and the PhD really unfolded, based on that sort of behavioural work. After that I fell into an academic position at Monash University, and the research just unfolded. We became, the lab became bigger and bigger. I started taking honours students, PhD students, started publishing work on the PhD, was attracting grants, and that sort of led to the path where I am now as a cognitive neuroscientist with an international platform of Huntington's disease research. Our lab really focuses on understanding the consequence of neurodegenerative disorders. In particular, we focus on understanding the progression of the disease and what factors influence the progression of disease, not only in Huntington's disease but in other neurodegenerative disorders such as Friedreich ataxia. So we use a range of platform technologies such as neuroimaging, in particular MRI, so we can discover how the brain changes and also look at consequences of behavioural changes, so we can understand not only the brain changes but how they're actually impacting the behavioural profile that we see in our patient groups. So Huntington's disease is a devastating neurodegenerative disorder. It targets the caudate and the putamen, and there is significant atrophy not only in these subcortical structures but also more widespread neuropathology. The main symptoms are problems with movements, voluntary movements, problems with cognition, as well as psychiatric disturbance. The clinical diagnosis of Huntington's disease, however, is made on unequivocal signs of involuntary movements. And these involuntary movements are known as chorea-type movements. Now, the word chorea comes from a Greek word meaning dance. So these involuntary movements, there's an excess of movement. The patient during the early stages of the disease can control these movements somewhat. However, they do become quite florid and quite hyperkinetic during the course of the disease. Now, the gene was identified in 1993 as a CAG triplet repeat, meaning that individuals with more than 39 CAG repeats will eventually develop symptoms. There is a single gene test that can confirm whether the individual is either gene positive or gene negative. The age at onset is 40 years of age, and the disease leads to eventual death around 15 to 20 years after the individual has been diagnosed. Huntington's disease is unique in many ways because of the fact that the gene has been discovered. It was discovered in 1993. So we can now study individuals who are at the pre-manifest stages of Huntington's disease. That is, they've had a gene test and they're either positive or negative. So it allows us to really study that window, that pre-symptomatic window, so we can understand what changes are going on in the brain well in advance of when patients do become symptomatic. And this is really critical because if we are to intervene with drug therapy, we want to try to intervene during that pre-manifest stage so we can forestall the onset of symptoms as much as possible. So therefore, if we look at the pool of neurodegenerative disorders, other disorders such as Alzheimer's disease or Parkinson's disease, even multiple sclerosis, the genes for those particular diseases have not been identified. Huntington's disease internationally is extremely well funded. There is an organisation in the US known as the CHDI Foundation where multi-million dollars are given to researchers around the world in order to accelerate the discovery of a cure for Huntington's disease. And so I think we've made significant advances in this disease over and above other diseases such as Parkinson's disease and Alzheimer's disease, although of course with those diseases there are some major discoveries that have been made as well. So Huntington's disease differs from disorders like, for example, Parkinson's disease. Huntington's disease is a young adult onset disorder, whereas Parkinson's disease, the age at onset is around 60 to 65 years of age. Both disorders are subcortical dementias. Parkinson's disease tends to affect the substantia nigra, pars compacta, whereas Huntington's disease tends to affect the caudate and putamen. The other major difference between Parkinson's disease and Huntington's disease is that the disease targets different pathways within the basal ganglia, and therefore Parkinson's disease is considered a hypokinetic disorder where you see a poverty of movement, you see a lot of bradykinesia, akinesia, as well as rigidity of movement, whereas Huntington's disease is more of a hyperkinetic disorder, that is, there's an excess of movement. You also see a little bit of bradykinesia as well as akinesia and rigidity toward the end stages of the disease, but these diseases selectively target different regions within the basal ganglia, and they therefore also target different excitatory and inhibitory pathways within the brain, which therefore lead to the differences between or in the symptoms that we see. So bradykinesia is a term that's used to suggest that individuals are having a problem in executing movements, whereas akinesia is a term that's defined by the fact that individuals have a problem in initiating movement. So when we conduct a motor program has both an initiation phase as well as an execution phase. So an initiation phase, that is how long it takes for us to initiate that movement, whereas whether it's lifting our arm to retrieve a glass of water, that phase is the initiation phase, how long it takes to initiate that move, whereas the execution phase is how long it takes for us to actually retrieve the glass. So therefore akinesia is defined by how long it takes us to initiate a movement, and bradykinesia is defined as how long it takes us to execute a movement. Neuroimaging technologies provide state-of-the-art tools to allow us to look inside the human brain. Using technologies such as MRI, diffusion tensor imaging, functional imaging, as well as other types of imaging methodologies such as transcranial magnetic stimulation or electrophysiology. It allows us to probe the brain and understand different tissue property. And this is really important in diseases such as Huntington's disease, because we're still to determine whether the structural changes in the brain occur prior to the functional changes, and so using cutting-edge imaging technology provides an important window to allow us to understand some of these fundamental changes that are occurring very early in the human brain. So when we obtain an MRI, we can assess structure of the brain. So this allows us to look at whether certain structures in the brain are changing in volume over time. Diffusion tensor imaging allows us to look at microstructural changes in the brain. We can start to look at white matter changes. So we're getting down to a more micro level of the property of the neuron in a way that a structural scan doesn't allow us to. Functional imaging allows us to assess how different regions of the brain are working whilst the patient is performing a cognitive task. So it allows us to assess the blood oxygen or the blood that's been recruited in those particular regions in order to help the patient stay on task. So it doesn't offer insight into volume or microstructure, it offers insight into brain function. Now, with imaging technologies and with the advance in various computational methods, we're able to study the brain as a network. So we've got all our scans, so we can apply different tools and different methods to start to analyse the brain as a network. So we can actually tell the software that we're interested in, a couple of cortical structures and a couple of subcortical structures, and therefore the analysis will spit out the connectivity or the degree of strength in the connections between those particular regions. And that is absolutely critical if we're looking at diseases such as Huntington's disease, where we can start to use advanced computational methods to look at how the brain, how the network, how the circuits in the brain change over time. So IMAGE-HD is an internationally funded study that's run at Monash University. It's a multimodal imaging study looking at a group of pre-manifest individuals who are gene positive, as well as a group of symptomatic patients who have been diagnosed with Huntington's disease, and a group of age-matched healthy controls. It's a 5-year study, and the primary aim is to look at disease progression using imaging technologies. So we've studied our individuals at baseline, at 18 months follow-up, and again at 30 months follow-up. Because it's a multimodal neuroimaging study, we're acquiring brain scans looking at structural changes with MRI. We are also obtaining diffusion tensor images to look at connectivity in the brain, and we also have a functional imaging protocol. One of the major findings from IMAGE-HD is that we're able to document structural changes in the brain that are occurring very early in the disease, even up to 15 to 20 years prior to when individuals become symptomatic. So these volumetric changes are suggesting that the brain is changing in the absence of any symptoms. The patients are able, or the individuals who are pre-manifest are working in the community, they're undertaking their day-to-day activities. However, there are structural changes that are occurring in the brain. Our functional imaging data has shown us that in addition to these structural changes that are occurring very early, that the brain has a capacity to reorganise itself. So what we've found with a working memory task is that cortical and subcortical areas of the brain are showing increased activation during these pre-manifest stages, whereas we don't see that in normal controls. So this is suggesting that the brain is able to rewire itself or tap into areas of either cognitive reserve or tap into areas to increase the level of activation as a consequence of these structural changes that we're seeing very early in the disease. So this tells us that the brain has an extraordinary capacity to rewire itself, to tap into areas of cognitive reserve, to compensate in a way that will allow the patient or the individual to stay on task and to perform day-to-day activities until such time where that capacity starts to decline, where the individual becomes symptomatic and they're unable to tap into those cognitive reserves in order to help that individual stay on task. So therefore, with symptomatic Huntington's disease, we do see a sharp decline in cognitive function. So I think this provides us with an opportunity to look at whether we can introduce certain strategies in order to help participants stay on task. There's a lot of interest now in cognitive training, in physical training, non-pharmaceutical interventions that may be of benefit in forestalling the onset of the disease. So I think there's a lot of interest in this area, and I think it's yet to be determined as to whether these particular training opportunities will have a benefit on forestalling the onset of the disease. But I think we know enough from the ageing literature and from other disorders such as Alzheimer's disease and other dementias where cognitive and physical training has certainly made a difference to the overall cognitive profile of the individual, and in some cases it has been linked to volumetric changes in the brain. Longitudinal studies are absolutely critical if we are to document the disease progression and look for sensitive biomarkers to track progression. Biomarkers can be brain measures, they can be clinical measures, they can be cognitive measures, or any other measure that tracks the trajectory of progression. Biomarker discovery is absolutely critical for clinical trials, because if we find that a particular measure is sensitive to progression, that is, it shows the largest effect size, we can recommend that to clinical trials so that that measure can be incorporated into such studies, so we can look at the efficacy of how that drug is effective in forestalling further deterioration of that particular measure. Cross-sectional studies only give us only provide an opportunity to look at one time point or one moment in time, whereas a longitudinal study allows us to repeat our measures over multiple times so we can assess how that particular measure deteriorates over time. And certainly we know that some measures are more sensitive to disease progression than others. So by providing a battery of tests, whether they are imaging measures or neurocognitive, neuropsychiatric measures or clinical measures, we're increasing the repertoire of possible sensitive biomarkers in terms of discovering which are the most sensitive in tracking disease progression, and therefore making those recommendations to drug companies for future clinical trials. So the current treatments for Huntington's disease involve neuroleptic medication, which control to some extent the involuntary movement disorder in patients, as well as antidepressant medication to control the depressive and anxiety symptoms that are also a consequence of the disease. Currently, however, there remains no cure for the disease. There are currently a number of clinical trials that are run worldwide. We're looking at new compounds that could forestall the onset of disease, but it's still very early days in order to be able to say definitively that these particular trials will move to phase 3, phase 4, phase 5 of clinical trial testing. Currently, clinical trials are only on symptomatic Huntington's patients, but we do hope in the future clinical trials will involve pre-manifest individuals, because we know that people are pre-symptomatic, free of symptoms, until such point in time where decline starts to occur. So we want to extend that pre-symptomatic window as much as possible by intervening with a drug compound early in the early stages. However, as I mentioned, currently clinical trials are only with symptomatic individuals. We want to test the efficacy of such trials, go through the steps before we can feel confident that the compound is safe, it's having an effect, and then look at how it can be introduced in pre-manifest individuals. It's still very early days to understand how stem cell therapy could potentially be used in diseases such as Huntington's disease. They have shown some promise, however there are still a lot of controversies surrounding stem cell therapy, so it is look at this space for the future. So my next steps as a researcher is to understand brain compensation and delve into that in more detail. So we do have an interest in pursuing non-pharmaceutical interventions, things like whether cognitive training or physical training has the capacity to tap into some of this cognitive reserve or into the compensatory mechanisms that we've been able to define through our IMAGE-HD study, and to understand how the brain can change itself or rewire itself as a consequence of cognitive training and physical therapy. We see this in our lab as the future for diseases such as Huntington's disease. We know with clinical trials it can take up to a decade for a positive result to become known. So non-pharmaceutical intervention offers almost an immediate way of looking at how we can best change the course of the illness.